In this video, we'll be discussing about the adipocyte differentiation via signaling pathways. We see there are three pathways that regulate the adipocyte differentiation. First one is the AKT PKB signaling pathway. Second one is the Wnt signaling. And third one is the TGF-β signaling. The AKT PKB is the stimulatory pathway for adipocyte differentiation, whereas Wnt and TGF-β are both the inhibitory pathways for adipocyte differentiation. The adipocytes are the fat storing cells called the lipocytes that constitutes the adipose tissue. And this tissue gets differentiation from mesenchymal stem cells in the form of preadipocytes first. Now let's get to the molecular level. We see we have the PPARγ, which is the transcriptional factor in the cells. It regulates the adipocyte differentiation. This PPARγ acts as a master regulator of adipocyte differentiation along with EBPα, which is another transcription factor that induces adipocyte differentiation. Now let's get directly to the signaling pathways. If we see the structure of cell, we see we have the cell membrane first. It's having RTK receptor in the form of monomers. Then it has Fresnel receptor, and also it has got TGF-β receptor, which is formed from type 1 and type 2 receptor. So we have these three receptors on the cell membrane, and it is these three receptors from which we are going to see the signaling pathways for adipocyte differentiation. Then on the lower side we have the nuclear membrane, and inside of the nucleus we have DNA molecule which is having different sequence for different genes, and of those we have PPARγ gene, and we also have EBPα genes. Both the genes transcribe and translate into respective proteins. It's these genes that regulate the adipocyte differentiation. Both these genes translate into adipose induction genes, which we are going to see later on in this video. Moving on to the AKT PKB signaling, first we see we have two factors in the cytoplasm, Necdin and GATA2. Both these factors are negative factors for adipocyte differentiation. Necdin and GATA2 inhibits the transcription of PPARγ factor, which then inhibits the adipocyte differentiation, as PPARγ induces the adipose genes. So when PPARγ is inhibited, that means adipose genes are not getting translated. So at first we need to stop or inhibit these two factors, Necdin and GATA2, and this is done by AKT PKB signaling. We see when the insulin binds the RTK monomers, it induces dimerization of RTK receptor, with which the tyrosine residues of RTK are autophosphorylated as shown in the diagram. The RTK activation or its phosphorylation then recruits the IRS towards the receptor, which also gets phosphorylated here. Then furthermore, P85 subunit of PI3 kinase is recruited to the receptor, which ultimately takes in the P110 subunit of PI3 kinase. So from this event we get the formation of active PI3 kinase, which is P85 plus P110 subunit. Now this PI3 kinase works on Necdin and GATA2 and inhibits both of these factors. So when these factors are inhibited, the PPARγ remains active without any inhibitory signal. And eventually we get the adipose gene induction and the adipocyte differentiation stimulation. But the other two pathways are inhibitory pathways. We see Wnt binding towards the Fresnel receptor induces the activation of β-catenin, which in turn activates the TCF, and this TCF has ability to downregulate the transcription of PPARγ and CEBP transcription. On the other hand, the TGF-β binds its receptor and activates the SMAD3. And this SMAD3 inhibits the forward loop of CEBP factor towards the PPARγ. So what we get from these complex mechanisms? We see in the presence of insulin, the Necdin and GATA2 are inhibited, with which master regulator of adipocyte differentiation, that's PPARγ, continues its transcription. That in turn stimulates induction of adipose genes, so we get adipocyte differentiation. And also CEBPα stimulates the same positive signal for adipocyte differentiation. So this is how adipocyte differentiation takes place through signaling. I hope you like the video. If you like it, give it a thumbs up. Do consider supporting my work on Patreon, and also make sure to subscribe this channel. Thanks.